Okay, this is chapter 3 of Clinical Electrophysiology. It's on electrotherapy and electrophysiologic testing. So I'm going to go through and read some of this chapter. Electrical stimulation has many therapeutic applications. Currently and historically, clinical electrical stimulation has been used primarily to activate electrically excitable tissues, nerve, and skeletal muscle. The appropriate use of electrical stimulation in therapeutic applications requires the clinicians have a clear understanding of the basic structure and function of these tissues, including the mechanisms of their activation by the central nervous system and electrical currents. The primary objective of this chapter is to review the physiology of skeletal muscle and peripheral nerve as it applies to clinical applications of electrical stimulation. The effects of electrical stimulation on other tissues, such as skin, are addressed in Chapter 8. Electrical excitability of muscle and nerve, membrane structure, and the resting membrane potential. Structurally, the cell membranes of muscle and nerve cells are similar to those of other cells, consisting of phospholipid bilayer containing a variety of protein molecules. The proteins in nerve and muscle cell membranes, however, serve several special functions that set these tissues apart from other body tissues. Membrane proteins may serve as receptor proteins, which act as binding sites for neurotransmitters or neuromodulators. Channel proteins, which under certain conditions form a pore through the membrane for the movement of ions, such as sodium, potassium, chloride, or calcium. And C, transport proteins, which bind and transfer substances such as sodium and potassium through the membrane, often against concentration gradients. Each of these types of proteins is thought to undergo a change in shape, conformational change, as they perform their respective functions. By virtue of its structure, the excitable cell membrane functions as a barrier to the movement of substances between the intracellular and extracellular spaces. Substances such as oxygen, carbon dioxide, and water move readily through the phospholipid bilayer. In contrast, ions and macromolecules such as proteins do not move readily through the membrane. The excitable cell membrane has a differential permeability to different electrical charged particles. In the resting state, the nervous system is not activating excitable tissue. Excitable membranes are readily permeable to potassium ions, slightly permeable to sodium ions, and impermeable to a number of large negatively charged proteins and phosphates, or anions. Because a large number of anions are trapped within the cell and the membrane is readily permeable to potassium, the positively charged potassium ions are drawn into the cell by electromotive forces as the resting state is being established. Another important property of excitable membranes in the resting state is that they use cellular energy in the form of adenosine triphosphate, or ATP, to actively transport sodium and potassium across the membrane. The active sodium-potassium pump moves sodium ions out of the cell while moving potassium ions in. An important feature of the sodium-potassium pump is it is capable of moving the ions against the electrochemical forces tending to oppose their movement in the resting state. As a consequence of both the selective permeability of the membrane and the membrane pumping mechanisms, the distribution of charged particles across the membrane of electrically excitable cells is not uniform. The concentration of sodium is higher in the fluid surrounding the cells, and the concentration of anions and potassium is higher inside. The resulting separation of electrically charged particles gives rise to an electrical potential difference across the membrane. This transmembrane potential difference or voltage is referred to as the resting membrane potential, and hence the excitable cell is described as polarized. The resting membrane potential, or RMP, exists all along the length of the nerve or muscle cell. For nerve cells, the RMP exists across the membrane cell body, dendrites, and the entire length of the nerve cell axon. The magnitude of the resting membrane potential is approximately -90 millivolts for muscle and is slightly less, about -70 millivolts for peripheral nerve fibers. The absolute RMP values for muscle and nerve of different species vary. Under normal conditions, as long as the membranes of these cells remain intact and ATP can be supplied to the sodium and potassium pump, the resting membrane potential of these cells remains stable. By virtue of the concentration differences of sodium and potassium ions across the membrane and the relative negative charge within the resting membrane, both the chemical and electrical forces acting on sodium are directed into the cell, while the net electrochemical forces acting on potassium are directed out of the cell. Action potentials. Muscle or nerve cells are different from other types of body cells because of specialized cell membrane properties that enable them to initiate and propagate action potentials. What are action potentials and why do they occur? In contrast to the membranes of non-excitable cells, muscles and nerve cell membranes may quickly and dramatically alter their permeability to ions in response to chemical, electrical, thermal, and mechanical stimuli. When appropriate stimulus is applied to a small region of the excitable cell, for example, the peripheral nerve fiber, the membrane permeability to sodium and potassium ions transiently increases. Because both the concentration and electrical forces for sodium are directed inward, sodium ions move into the cell and the transmembrane potential is reduced or approaches zero. This reduction in transmembrane potential, referred to as depolarization, is gradual at first and a small amount of sodium leaks into the cell. When the transmembrane potential reaches a critical voltage level called the threshold, voltage-sensitive sodium and potassium channel proteins in the membrane undergo a conformational change and open. Permeability to sodium abruptly increases as sodium gates open, whereas permeability to potassium increases more slowly because the electrochemical forces acting on potassium ions are initially much lower than those acting on sodium ions. Because both the electrical and chemical forces acting on sodium are initially directed strongly into the cell, sodium rushes in when sodium channels open. As a result, the transmembrane potential rapidly depolarizes as positively charged sodium ions are added to the negatively charged cell interior. If sodium channels remain open for very long, sodium ions would continue to enter the cell until electrical and chemical forces acting on sodium ions were balanced. The sodium equilibrium potential, which would occur approximately at plus 60 millivolts. In fact, sodium influx stops at a point when the transmembrane potential reaches at about plus 35 millivolts, because at this level the sodium channels close. At this voltage, the membrane again becomes relatively impermeable to sodium. When the sodium gates close, the potassium gates remain fully open. Because inside the cell is now positively charged and only a few potassium ions have left the cell, both the electrical and chemical forces acting on the intracellular potassium ions strongly force potassium ions out of the cell's interior. The potassium gates remain open, so positive ions rush out of the cell, thereby removing positive charges from within and the transmembrane potential becomes progressively more negative. Potassium efflux from the cell quickly makes the transmembrane potential negative again, in a process called repolarization. The forces moving potassium out of the cell are so great, and the potassium gates remain open long enough that the membrane is repolarized slightly past the resting membrane potential, about 10 to 20 millivolts below the resting membrane potential. This hyperpolarization of the membrane proceeds until no net electrochemical forces act on potassium. The transmembrane potential at which no electrochemical forces exist to cause potassium ion efflux is called potassium equilibrium potential. At that point in time, potassium gates continue to close, and passive diffusion of ions across the membrane then quickly restores the membrane potential to its original resting level. The transmembrane voltage changes that occur in response to excitable cell stimulation are collectively called an action potential and are generally completed within 1 microsecond. These voltage changes occur in response to each stimulus sufficient to raise the transmembrane potential to threshold level. The voltage changes of the action potential are simply a reflection of the sodium and potassium currents that occur over time across the membrane in response to the opening of the sodium and potassium gates. If an excitatory stimulus is large enough to depolarize the membrane to its threshold level, the transmembrane voltage changes that occur by virtue of ion movement happen in precisely the same manner for each stimulus applied. Because the amplitude of voltage changes in response to stimulation is constant from stimulus to stimulus, the AP is described as all or none. The AP action potentials that occur in nerve and muscle cells in response to either normal nervous system activation, the application of sensory stimuli, or electrical stimulation are basically similar. In monitoring the normal function of peripheral nerve and muscle, single isolated action potentials are rarely produced. Usually, volitional activation of nerve and muscle produces series of action potentials in these cells. The frequency of action potentials produced in nerve and muscles during normal activities rarely exceeds 60 per second. And ordinary discharge ranges from 5 to 15 impulses per second. In contrast, during electrical stimulation of excited cells, the action potential frequency mirrors the stimulation frequency up to a maximum rate of near 1,000 impulses per second in nerve fibers. This maximum rate of action potential generation is determined by the period following the onset of the potential during which the nerve or muscle fiber is absolutely or relatively unexcitable, called the absolute or relative refractory period. Where are action potentials initiated? Under normal physiological conditions, action potentials in multipolar neurons, such as those innervating skeletal muscle, begin at the axon hillock, the point at which the axon rises from the cell body of the neuron. In peripheral sensory neurons, action potentials are normally initiated at the peripheral or distal end of sensory axons, often the point where the axon terminates in a specialized receptor ending. In skeletal muscle fibers, action potentials are initiated in the region of neuromuscular junction between the alpha motor neuron and the muscle fiber. Of importance in clinical electrophysiology, action potentials may be initiated using electrical stimuli at any point along the course of the peripheral nerve axons, where the electrical stimulus is sufficient to trigger the opening of voltage-gated sodium and potassium channel proteins. Action potential propagation. Although the opening of sodium and potassium gates are resultant action potentials have been discussed as though they occur at one segment of the membrane, the channel opening and associated voltage changes spread to adjacent portions of the nerve or muscle membrane and trigger the same sequence of ionic movements across the membrane. In this manner, the action potential is said to propagate or be transmitted along the surface of the cell. The resultant transmembrane voltage changes, monitored in one portion of the nerve axon or muscle cell, will be identical to those recorded in any other region. The rate or speed of action potential propagation along membranes is not the same for all excitable cells and muscle fibers and nerve fibers of different diameters. The speed of action potential propagation increases as the diameter of the fiber increases. I'm sorry, I lost my place. You're heating me up. Okay. The differences in action potential, conduction, velocity, and excitable cells of different sizes are due to differences in the passive electrical properties among the various types of fibers. The two most important passive electrical properties with respects to the transmission of ionic currents in nerve and muscle fibers are the membrane resistance and the internal resistance. In response to electrical or chemical activation, current spreads within the fiber and becomes attenuated or reduced with distance. This attenuation occurs because ionic movement longitudinally along the axon is opposed by resistance and because some ions leak out of the fiber through its membrane. The higher in resistance or lower in membrane resistance, the less the spread of longitudinal currents along the fiber, and hence the lower the propagation speed of action potential currents. Small diameter fibers have relatively high internal resistance compared to large diameter fibers and consequently have lower conduction velocities than large fibers. In addition, large diameter peripheral axons have higher membrane resistance because they are covered along most of their length with an insulating layer of myelin, myelin. The transmission of action potentials along the membrane of both muscle or unmyelinated peripheral nerve fibers occurs in a continuous manner involving every segment of the membrane. Many types of peripheral nerve fibers, however, are surrounded by myelin, an insulating non-conductive material. The myelin layer is interrupted at intervals, nodes of Ranvier of about 1 millimeter, exposing small segments of the axon membrane to extracellular fluids. In myelinated axons, only those portions of the axon membrane in contrast with the extracellular fluid at the nodes of Ranvier undergo the permeability changes and ionic currents associated with the action potential. Ionic currents do not occur in those segments of the membrane covered by the myelin because the membrane resistance in those regions is extremely high, and areas of membrane beneath the myelin have relatively few voltage-gated channel proteins. When an action potential is initiated by electrical stimulation at one node, ionic currents spread longitudinally until they reach the adjacent node. As a result, current attenuation is less and impulse propagation is more rapid in myelinated axons than in unmyelinated axons. The AP currents moving along the axon membrane in myelinated axons can be viewed as moving in a stepwise manner from one node to the next. This form of action potential propagation in myelinated axons is called Syllatory Conduction, a term derived from the Latin verb saltare, which means to leap. The conduction velocity of myelinated nerve fibers is much higher than that of either unmyelinated axons or muscle fibers because myelinization reduces the amount of membrane involved in the ionic movements of the action potential. Hence, myelinization effectively reduces the amount of axon membrane through the ionic currents must flow. Action potential propagation occurs in neurons and at locations where voltage-gated sodium and potassium ion channel proteins are located. In multipolar neurons, the voltage-gated channel proteins are found on the single axon projecting from the cell body. Voltage-gated sodium and potassium channels associated with AP generation are not found on the cell body or dendrites of multipolar neurons. On unipolar or bipolar neurons, voltage-gated sodium and potassium channel proteins are located on both peripheral and central processes. Composition of peripheral nerves. Peripheral nerve fibers. Peripheral nerves link the spinal cord to tissues in the extremities and trunk, such as skeletal muscles, skin, joint capsules, vascular smooth muscle, and sweat glands. The primary components of peripheral nerves, such as the medium or sciatic, are the nerve fibers, axons. Nerve fibers are merely long, thin projections from nerve cell bodies that lie in or near the spinal cord or brainstem. Most peripheral nerves contain axons from three general types of nerve cells: motor neurons, first-order sensory neurons, and autonomic neurons. Because most nerve cells contain each of these components, most peripheral nerves are referred to as mixed. Some peripheral nerves contain axons originating from mainly sensory cells and hence are called sensory nerves. No peripheral nerves contain only motor or autonomic fibers. These cell bodies of motor neurons lie in either the ventral horn regions of the spinal cord or in brainstem motor nuclei of cranial nerves. The multipolar cell bodies of spinal motor neurons project a single axon through the ventral roots of the spinal cord and along the normal, well-established routes of peripheral nerves. All motor neuron axons terminate in muscle. Functionality motor neurons can be divided into two groups. Alpha motor neurons, the cells that innervate ordinary striatic skeletal muscle fibers, and gamma motor neurons, smaller multipolar cells that innervate in the special types of muscle fibers contained within a sensory receptor called the muscle spindle. The cell bodies of first-order sensory nerve cells in the dorsal root ganglia of the spinal cord or in selected cranial nerve sensory nuclei. The sensory neurons are either unipolar or pseudo-unipolar or bipolar cells that project one axon toward the spinal cord and a second axon toward the periphery. The periphery peripheral process of sensory neurons is the segment contained within the peripheral nerve. The peripheral axon of about half of the dorsal root cells terminate as free nerve endings in structures such as skin, muscle, and joint capsules. The remaining peripheral sensory axons end with some specialized type of sensory receptor, such as the Pacinian corpuscles, the Golgi tendon organs, or muscle spindles, which are designed to respond best to one type of sensory stimulus, such as touch. Muscle contraction force or changes in muscle length, respectively. The cell bodies of the autonomic nerve fibers and peripheral nerves are found in the chain ganglia that lie parallel to the spinal cord or in ganglia of the cranial nerves. The axon of autonomic neurons and peripheral nerves terminate in such places as the sweat glands and the smooth muscle and the walls of blood vessels. Under normal conditions of CNS activation, the motor neurons and autonomic neurons send action potentials away from the spinal cord and toward the periphery. Conversely, sensory cells of all types normally initiate action potentials in the periphery in response to stimuli and transmit the information to the spinal cord, whence it can be relayed to higher brain centers. With the primary functions noted, peripheral nerves can be viewed as simply action potential transmission pathways. Action potentials are carried along these two-way streets, and the transmission of signals in one direction along sensory fibers is not impeded by action potential transmission in the opposite direction along motor or autonomic fibers. The propagation of action potentials along axons in the normal or physiologic direction in a particular type of nerve fibers is referred to as orthodramic propagation. Orthodramic transmission for motor and autonomic fibers is away from the spinal cord, whereas orthodramic transmission of sensory fibers is toward the spinal cord. Although peripheral axons normally transmit action potentials either way, from or toward the central nervous system, all types of peripheral nerve fibers are capable of transmitting action potentials in both directions from point of initiation. This can occur, for instance, when electrical stimulation is applied at some point along the course of peripheral fibers. The transmission of APs in the direction opposite the physiological direction is called antidromic propagation. Antidromic action potential transmission in sensory cells, for example, is directed away from the central nervous system, whereas antidromic propagation in motor neurons is toward the central nervous system. The types of fibers in a peripheral nerve differ in size and structure. In general, the large cell body of origin, the larger the diameter of its projecting axon, and the higher the action potential conduction velocity. The differences in conduction velocity among the various types of fibers in a peripheral nerve have formed the basis for development of two commonly used nerve fiber classification schemes. The scheme developed by Gasser and colleagues is used to describe all types of axons: motor, sensory, and autonomic. In peripheral nerves, and the Lloyd scheme is restricted to the description of only sensory peripheral nerve fibers. Table 3.1 indicates the distributions of fibers, diameters, and conduction velocities for axons of specific type. The fiber diameters and conduction velocities were determined in animal nerves and generally exceed those of human peripheral nerve fibers. In spite of this, the relative difference in conduction speed of axons are believed to be similar in human nerves. Large diameter fibers innervating muscle spindle, Golgi tendon organ, and skeletal muscle conduct much more rapidly than the smaller diameter myelinated and unmyelinated pain efferents. Peripheral nerve is a composite of many tissues. Although the axons of sensory, motor, and autonomic fibers constitute the primary component of peripheral nerves, many other important types of tissue are contained within nerves. Structures that support peripheral nerve fiber function include Schwann cells, connective tissues, and blood vessels. All peripheral nerve fibers are surrounded by Schwann cells. They provide several important functions in peripheral nerve, including the formation of myelin. As indicated in the discussion of action potential propagation, approximately 1/2 of the peripheral axons in the average nerve are myelinated. Myelination, the segmented covering of many peripheral axons, increases the velocity of action potential conduction. In addition, Schwann cells insulate fibers from each other, which may explain why action potentials that are propagating distally in motor and autonomous fibers do not interfere with sensory action potentials traveling centrally. Peripheral nerve axons are also surrounded by 3 layers of connective tissue that serve several important roles in relation to nerve transmission. The Epi Neuron is a dense sheath of connective tissue that surrounds the entire peripheral nerve and projects inward to enclose and separate bundles of nerve fibers. One role of the Epi Neuron is to protect peripheral axons from compressive forces applied to the nerve. The perineuron is a thin sheath of flattened cells layered concentrically around bundles of nerve fibers. These perineural sheaths maintain intravascular fascicular pressure, act as a bidirectional diffusion barrier, and supply much of the elasticity and tensile strength or passive resistance to stretch of peripheral nerves. The innermost layer of connective tissue in the endoneuron, which is a fine connective tissue matrix surrounding every nerve fiber. The endoneural layer contains Schwann cells and endoneural fluid, which forms the extracellular environment of each nerve fiber. Peripheral nerves have a well-developed vascular supply. Longitudinally oriented arterioles and venules are present in the epineuron. These vessels regulate the blood flow to and from the peripheral nerve via sympathetic innervation of smooth muscle contained within their walls. At regular intervals, epineural arterioles branch toward the center of the nerve to supply blood flow to one or more nerve fascicles. Small diameter longitudinally oriented arterioles and venules are also found lying between cells of the perineuron. Branches arise and pass obliquely to form capillary beds within the fasciculus. The capillaries of peripheral nerves are relatively large in diameter, approximately twice the size of muscle capillaries, and are contained mostly in the endoneurium within the fasciculus. The intravascular capillary beds are characterized by extensive U-loop anastomosis between capillaries and provide several alternative pathways for blood flow to capillaries that supply the individual nerve fibers. Skeletal muscle structure. Muscle cell structure. The primary component of striated skeletal muscle is the muscle fiber. Muscle fibers are cylindrical, elongated cells that have diameters commonly ranging from 50 to 200 micro millimeters, and lengths that may reach many centimeters. These thread-like cells provide muscles with the electrical and contractile properties that are of special interest to those who study electrotherapy and electrophysiological evaluation. Occupying most of the interior of the muscle cells are bundles of specialized proteins called myofibrils. These cylindrical elements are 1 to 2 micromillimeters in diameter and are found throughout the entire length of the muscle fiber. A single large muscle fiber may contain as many as several thousand myofibrils. When a longitudinal section of muscle is viewed with the light microscope, a myofibril has a banded appearance with alternating light and dark areas. This striated appearance extends along the entire length of the myofibril. On closer inspection with an electron microscope, the structure of the myofibril becomes clearer. The repeating pattern of the myofibril is due to the arrangement of a system of contractile proteins, the thick and thin myofilaments contained within the myofibril. The repeating unit of the myofibril containing light and dark areas is called a sarcomere. A single sarcomere extends between structures known as Z-lines. Thick myofilaments are located in the center of each sarcomere, and thin myofilaments extend from each Z-line toward the center of the sarcomere and overlap the ends of the thick filaments. The dark bands of muscle are made up of thick filaments and overlapping thin filaments. The light bands contain only thin myofilaments extending from centrally located Z-line. Biochemical analysis has revealed that the thick filaments are composed of contractile protein myosin. This protein has a globular head connecting to a long, thin tail. Approximately 200 myosin molecules are arranged to make up a single thick filament. Thin filaments are primarily composed of the contractile protein actin. Actin molecules are spherical, and the thin filament is made up of 2 long chains of actin molecules, twisted together much like a twisted strand of pearls. Included on a thin filament are 2 other proteins, troponin and tropomyosin. These regulatory proteins control the interaction between the contractile proteins actin and myosin. Troponin binds free calcium ions and controls the position of tropomyosin. Tropomyosin is a large, thin protein that by the virtue of its position on the thin filaments, interferes with actin-myosin coupling in muscle at rest. Surrounding each myofibril in a sleeve-like fashion is a tubular network called the Sarcoplasmic Reticulum. The SR is a modified endoplasmic reticulum containing a high concentration of free calcium ions. The SR, Sarcoplasmic Reticulum, is divided into units along the length of the muscle fiber, and each unit is made up of 3 components. The central portion of each SR segment is called the fenestrated SR. The tubular network extending from the central region is called the longitudinal SR. The longitudinal SR connects to larger cavities called the lateral or terminal cisternae. The lateral cisternae from adjacent segments of the SR wrap around the tubules, which are invaginations, central projections of the surface of the cell membrane. Just beneath the surface of the cell membrane of each muscle fiber lie the elongated or ovoid nuclei of the cell. The nuclei along the length of the muscle fiber and are the storage sites for the genetic instructions for cellular activities such as the production of muscle protein. Since the muscle fibers are so long, it may be that many nuclei are required along the length to effectively regulate protein synthesis and degradation within the cell. Another set of internal structures located along the entire length of the muscle fiber are the mitochondria. Muscle fiber mitochondria are also known as sarcosomae, are rather evenly distributed throughout the volume of the muscle fiber and are commonly found lying between microfibrils. They contain enzymes associated with controlling the oxidative metabolism of muscle fuels, such as free fatty acids and glucose. Consequently, sarcomes are primary site of ATP production. ATP is required to supply energy for numerous intracellular processes, including protein synthesis, membrane pumps, and muscle contraction. The last major internal component of muscle cells, which surround all of intracellular structures, is cytoplasm. This intracellular fluid, also called sarcoplasm, is mostly water, but contains a variety of other substances critical to the normal function of the muscle cell. The sarcoplasm is the storage site for muscle fuels, including free fatty acids, glucose, and a number of other nucleotides like ATP. All of the enzymes of glycolysis are located in the cytoplasm, and hence the cytoplasm is the site where glycogen and glucose are anaerobically metabolized to produce ATP. The sarcoplasm is also rich in amino acids, which are the basic building blocks of proteins. Lysosomes are suspended in the cytoplasm for handling of damaged intracellular components. Finally, the cytoplasm contains myoglobin, an oxygen-binding and transport protein, which is essential for the maintenance of oxidative metabolic processes. The cell membrane of the muscle fiber is called the sarcolemma. The membrane regulates the substances that are allowed to enter and leave the cell, maintains the muscle fiber's resting membrane potential, and propagates muscle action potentials as described previously. The specialized region of muscle fiber membrane where action potentials are normally initiated is called the motor end plate. At the motor end plate, the axon of the nerve cell innervating the muscle fiber, the alpha motor neuron terminates. This region of the muscle cell contains specialized receptor proteins for the neuromuscular junction transmitter, acetylcholine. When acetylcholine is released from the motor neuron terminal and binds to receptors at the motor end plate, the cell membrane in the region becomes more permeable to sodium ions. So many sodium ions leak into the cell that a muscle action potential is initiated each time the motor neuron to the muscle is activated. The normal neuromuscular junction is therefore often described as an effective synapse. Another special feature of the sarcolemma is that at regular intervals, the cell membrane is invaginated to form channel-like structures called T-tubules. Which dive toward the center of the cell. The lumen of each T-tubule is continuous with extracellular fluid, and the T-tubules provide a pathway for action potential propagation to the center of the muscle fiber. Muscles as a composite of many tissues. In a discussion of the structural features of muscle, it is common to focus primarily on the organization of muscle fibers, groups, and on the morphological features of individual muscle fibers. Although this is certainly valuable as a basis for understanding, another often overlooked perspective is that of the whole skeletal muscle as a composite of many different types of tissue. Certainly, the primary component of muscle is the muscle fiber or muscle cell. Fiber contraction gives whole muscle the ability to generate and sustain force used to either move or stabilize the skeleton. Several other types of tissue, however, are present within the whole skeletal muscle that may significantly influence the muscle's ability to generate or transmit force. One important component of whole muscle is connective tissue. The connective tissue within the whole muscle is subdivided into three main segments. First, epimysium is the dense layer of connective tissue that surrounds the entire muscle. Second, the perimysium is the layer of connective tissue that extends from the epimysium and surrounds groups of muscle fibers called fascicles. Fascicles. Finally, the endomysium is the fine, lacy, delicate layer of connective tissue extending from the perimysium and composed of collagen fibers and fibroblasts that closely invest each muscle fiber. These three layers of connective tissue account for much of the muscle's resistance to passive stretch. Changes in either the character or amount of connective tissue in muscle can dramatically alter the way in which the contraction force is transferred to the skeletal system. Another important secondary component is the vasculature of skeletal muscle. As they penetrate the epimysial connective tissue layer, large arteries rapidly branch into complex arteriolar networks in the perimysial layer. The arterioles in muscle are the main blood vessels regulating muscle circulation, and they branch to form elaborate capillary beds. In skeletal muscle, each fiber is served by at least one and usually several capillaries that lie next to the cell membrane in the endomysial layer. At the capillary muscle cell interface, exchange of fuels and metabolic substrates and byproducts occur. The capillary networks condense to form venules, which drain into large veins, which in turn exit muscle at the point where the arterial supply entered. The circulatory network of muscle is vital to normal function of this tissue because muscle is one of the body's largest consumers of oxygen and energy. Without an efficient operating vascular supply, muscle rapidly loses its ability to sustain contraction, as reflected in weakness and low fatigue resistance, often seen in individuals with circulatory disorders of muscle. A third component of whole muscles consists of nervous tissue. The skeletal muscles contain both myelinated and unmyelinated nerve fibers. Myelinated nerve fibers innervate not only skeletal muscle fibers, but also numerous types of sensory receptors that monitor muscle activity. Sensory receptors include spindles that monitor muscle length, rate of change in muscle length, and under certain conditions, muscle force. Gogli tendon organs are receptors at musculotendinous junction that monitor the active tension generated by muscle during contraction. In addition, muscle contains numerous free nerve endings that are terminations of either myelinated or unmyelinated nerve fibers associated with the detection of muscle pain. Other nerve fibers contained within muscle include the larger myelinated axons of alpha motor neurons, which, when activated, cause the skeletal muscle fibers to contract, and the smaller size of axons of gamma motor neuron system, which regulate the sensitivity of muscle spindles. The remaining nervous system structures and muscles are the fibers of the autonomic nervous system that are primarily involved in the control of muscle circulation by the regulation of vascular smooth muscle in the walls of arterioles and venules. All of the neural elements within the whole muscle play important roles. The alpha motor neuron axon endings in muscle are essential to initiate muscle contraction, as detailed in the following section of this chapter. The sensory receptors in muscle and the sensory afferent fibers that transmit their information to the brain and spinal cord provide valuable information to the central nervous system regarding muscle position and the state of contraction. Such sensory feedback is essential to the regulation of muscle contraction, and hence to the control of posture and movement. Dysfunction of neural components of whole muscle may lead to a reduction in the muscle's ability to produce levels of contraction appropriate to the demands of routine activities. Physiology of muscle contraction. Initiation of contraction. The series of electrical and chemical events that describe how skeletal muscle is brought to contraction is referred to as excitation-contraction coupling. To activate muscle, the central nervous system first initiates action potentials in the axons of alpha motor neurons. Once initiated, the nerve AP passes rapidly along the peripheral axons and finally sweeps over the membrane of the motor nerve terminals. As the nerve action potentials invade the terminals, acetylcholine, the transmitter substance at the neuromuscular junction, NMJ, is released into the fluid in the region of motor end plates through the process called excitosis. The acetylcholine moves by diffusion across the gap between the nerve and muscle membranes. When the transmitter reaches the muscle cell membrane, it is rapidly bound to specialized receptor proteins in the end plate region of the membrane. Immediately on the binding of acetylcholine, the membrane of the region of the receptors increases its permeability to sodium ions by opening sodium channel proteins. Because both electrical and chemical gradients for sodium ions are directed inward, sodium ions move into the muscle cell and the transmembrane muscle potential is reduced, or becomes less negative, as positive charges enter the muscle cells. This reduction in transmembrane muscle potentials can be recorded as an end plate potential. In normal muscle, the end plate potential produced in response to the transmitter released from the motor nerve terminal is always great enough to increase the sodium permeability of the muscle fiber membrane just outside the NMJ, thus triggering a muscle action potential. Sodium ions rush into the muscle, followed by potassium ions flowing out of the cell in a regenerative process that quickly sweeps over the surface of the entire muscle fiber. See previous discussion on resting membrane potential and action potential for further details. As the process of the muscle action potentials pass over the membrane, they encounter the invaginations of the surface membranes called T-tubules. The action potential currents invade the T-tubules and are carried out along the membrane toward the interior of the cell. As the depolarization is carried along the T-tubule, it triggers the opening of voltage-sensitive calcium channels and hence an increase in permeability of the lateral cisternae of Sarcoplasmic Reticulum to calcium ions. Because the Sarcoplasmic Reticulum contains a high concentration of calcium ions in comparison to the muscle cell cytoplasm, calcium rapidly diffuses into the cytoplasm. Once released into the sarcoplasm, calcium diffuses away from the sarcoplasmic reticulum into the region of the thick and thin filaments. On the surface of the thin filaments lies the regulatory protein troponin, which has a very high affinity for calcium ions. Any free calcium in the cytoplasm rapidly binds to any open calcium binding site on the troponin molecule. When calcium binds to the troponin molecules on thin filaments, a conformational change of troponin is produced. Troponin is attached to another protein, tropomyosin, as the troponin changes shape, and so it pulls on the tropomyosin and causes an alteration in the position of tropomyosin on the thin filament. Normally, tropomyosin is located in the position on the thin filament covering the site on each actin molecule that has a high affinity or attraction for the heads of the myosin molecules extending from the thick filaments. As troponin draws the tropomyosin away from these binding sites, actin molecules and myosin heads quickly attach. This attachment of myosin heads to the binding sites on actin. Cross-bridge formation immediately triggers a change in shape of the myosin molecules. In effect, the myosin heads swivel, and in doing so, draw the thin filament toward the center of the sarcomeres. This interaction between actin and myosin, which occur at literally thousands of sites, is the process that produces the force of muscle contraction. Cessation of contraction. The interaction of actin and myosin at any available site will continue as long as sufficient amounts of ATP are available to fuel the swiveling of myosin, as long as the binding sites of the actin molecule remain unblocked. In response to single surface muscle action potentials, a pulse of calcium is released. Some of the calcium binds to troponin, cross bridges between actin and myosin are rapidly formed, and muscle force generation begins. Almost as soon as it is released from lateral cisternae, calcium is actively transported back to into the SR by ATP-dependent pumps that lie in the wall of the longitudinal cisternae. As the concentration of free calcium falls into the cytoplasm, other bound calcium ions dissociate from the troponin. The troponin thus returns to its original shape, and the tropomyosin falls back into its rest potential, where it blocks acting binding sites for myosin. Very rapidly, the number of cross bridges that can form is reduced, and the force of contraction falls. When all acting binding sites are blocked by tropomyosin, no thick and thin interaction is possible and contraction ceases. Factors influencing muscle force production. Frequency of activation. Figure 3.12 shows the force produced within a segment of muscle in response to activation by a single AP when the fibers are held at constant length. The force produced reached its maximum value almost instantaneously and then gradually falls back to zero. The force pattern generated is a reflection of the rapid formation of many actin and myosin cross bridges within the muscle, followed by the gradual reduction in the number of cross bridges as calcium is gradually resequestered into the SR, and myosin binding sites on actin are covered by the tropomyosin regulatory proteins. SR is Sarcoplasmic Reticulum. Although in theory this force profile is produced within the muscle segment, the force that can be measured experimentally in response to a single activation is very different in character. Figure 3.12 also shows the isometric force measured from a segment of muscle in response to a single stimulus. In contrast to the force produced within the muscle segment, the force actually measured from the segment rises gradually to an amplitude that is less than produced by internal contractile apparatus. This force measured over time in response to a single activation is called isometric twitch. For mammalian skeletal muscles, the peak in twitch tension is reached about 200 to 100 microseconds after the force begins to rise. Why is the form of the isometric twitch different from that predicted for the internal contractile apparatus of muscle? The answer lies in the way in which the contractile machinery is harnessed to the skeletal system. As discussed previously, the contractile proteins of muscle that produce its force are encircled by elements such as Sarcoplasmic Reticulum, the muscle cell membrane, and the layers of connective tissue throughout the muscles. The layers of connective tissue within the muscle segment in turn merge at each end of the muscle to form the tendons of the muscle. Each of these structures and possibly others are elastic in nature. That is, they possess a spring-like quality such that applied forces stretch them, but when the force ends, they return to their original size and shape. The force produced within a muscle segment is modified, reduced in amplitude, and delayed in time by the presence of these muscle components, which may account for the form of the measured isometric twitch. The contractile elastic elements of muscle can be modeled as shown in Figure 3.13. The force produced by the contractile elements of muscle must be transmitted through the spring-like tissue elements called the series elastic components of muscle before it can be measured. When the contractile elements of muscle are activated, some of the force produced is used to stretch out this series elastic components, while the remainder is transferred through the skeletal system. The amount of force is used to stretch out the SEC is equal to the difference in peak of the internal force produced by and the peak in the isometric twitch tension. The time difference between the contractile element peak force and the peak in measured isometric twitch is due to the fact that some time is required to stretch the SEC. In comparison of the contractile element and twitch forces reveal that twitch tension is a poor indicator of the muscle segment's true ability to generate force. Thus far, the consequences of muscle activation by only a single isolated stimulus have been described. During the natural activation of muscle, however, a series of muscle action potentials is usually produced on the muscle cell membranes. How does such a train of action potentials influence the force produced by the muscle? In response to the first muscle action potential of a train, the contractile element of muscle is activated, and the force of the twitch-like contraction is produced. If a second action potential is delivered before all the calcium released by the first action potential is resequestered, more calcium from the sarcoplasmic reticulum is released, and action myosin cross bridges cycling is allowed to continue. As a result, the force measured from the activated muscle segment rises again. Since the SEC stretches in response to the first AP and it does not have time to fully relax before the second AP, less of the tension generated in response to the second activation is required to stretch out the SEC. Consequently, more tension is transmitted through the spring-like harness and can be measured. Subsequently, action potentials in the train produce even further increase in the measured force output of the activated tissue. The profile of force output that appears with oscillations in the tension record is called unfused tetanus. Unfused tetanic contractions are generally elicited in response to activation of the muscle at frequencies from approximately 3 to 20 stimuli per second, depending on the particular type of muscle activated. As the frequency of the skeletal muscle activation is increased, the isometric force can be measured from muscle also increases. In addition, the oscillations in force gradually disappear and the contraction becomes progressively smoother. Smooth, strong contractions produce in response to very rapid delivery, less than 20 to 30 per second of action potentials to muscle is state called fused tenatus. Maximum fused tetanic contractions are better indicators of a muscle's contractile capabilities than are twitch contractions. We're going to take a quick intermission, 5 minutes. Okay, end of intermission. Number of fibers activated. A second important factor associated with the regulation of muscle force is the number of muscle fibers activated in a contraction. As the percentage of total muscle fibers activated by motor nerve fibers is increased, the amount of force produced by the muscle at the fixed frequency of activation is increased. The increase in force as greater numbers of muscle fibers are recruited into contraction simply results from increasing the total number of actin-myosin cross bridges formed. Length of skeletal muscle. A third factor that determines the active tension that a muscle can produce is the muscle length. The length of the muscle at which the maximum number of cross bridges between thick and thin filaments can be formed is called rest length. For many human limb muscles in situ, muscle length can be altered by 20% of rest length. For example, when the elbow is fully extended, the biceps will be stretched to a length 20% greater than that in the mid-range. Conversely, if the elbow is fully flexed, the biceps length will be about 20% less than the rest length. Muscle generates the maximum amount of tension in the 10% range of rest length. In biceps, for example, the maximum tension is generated by the muscles in the middle 50% of the elbow's range of motion. If the biceps is either lengthened or shortened, such as elbow position in the middle of 50% of its range of motion, biceps tension generated capacity is reduced. Up to this point, muscle contraction has been discussed only under isometric conditions when the muscle is held at constant length. If during activation the muscle is allowed to change length, the velocity of shortening becomes a determinant of the amount of force the muscle can generate. Figure 3.15b shows the force-velocity relationship for concentric shortening isometric and eccentric lengthening contractions for skeletal muscle. The graph reveals that the higher forces are produced by muscle under isometric conditions when the velocity of shortening is 0, and the progressively declines as the speed of shortening is increased. The graph also illustrates that the highest force levels that a muscle can produce occur in eccentric contractions. Non-homogeneous nature of skeletal muscle. Skeletal muscle fiber characteristics. In previous description of the structural and contractile characteristics, skeletal muscle was treated as if all muscles were identical. In fact, it has been known for several hundred years that several different types of skeletal muscle exist, and that the differentiation of skeletal muscle appears to be related to how the muscles are used in normal activity. A classic comparison is made between the muscles of the birds of flight and those of domestic fowl. The breast musculature of chickens is pale in comparison to the dark leg muscles. These barnyard animals rarely use the breast muscles, which move their wings, but use the leg muscles the majority of their waking hours while foraging. In contrast, the breast musculature of migratory birds is dark, while leg muscles are paler in appearance. Sipp's observational findings led early scientists to suspect that there were two types of muscle fibers, light and dark, that made up skeletal muscles. With the advent of microscopic and histochemical techniques in the early and middle part of the 20th century, we learned that most mammalian skeletal muscles are composed of not two, but of three types of muscle fibers. Over the years, two major classification schemes have developed to describe the different types of fibers within mammalian striated muscles. The percentage of these fibers that make up muscles varies from muscle to muscle and from species to species for analogous muscles. In humans, the muscles of the extremities are frequently composed of approximately half fast twitch muscle fibers and half slow twitch fibers. Basic functional element of the neuromuscular system, the motor unit. From a structural perspective, skeletal muscle is composed of single muscle fibers of three different types. From a functional perspective, however, muscle contraction does not occur by the isolated activation of single muscle fibers. During normal activities, the force of muscle contraction is produced by the activation of muscle fiber groups acting in concert in response to a stimulus provided by the single nerve innervating each fiber in the group. A motor neuron, along with the group of muscle fibers it innervates, is called a motor unit. The cell body of multipolar neuron of the motor unit is located in the ventral horn of the spinal cord or in cranial nerve motor nuclei. For motor neurons arising from the spinal cord, large diameter myelinated axons project from the cell body, pass through the ventral root of the spinal cord, and subsequently run peripheral nerves until they reach the target muscle fibers. As motor axons enter muscle, they divide many types into fine branches, which in turn innervate single muscle fibers. The muscle fibers of a single motor unit are usually distributed over a rather large portion of the cross-sectional area of the muscle, and all of the fibers of a motor unit are of the same histochemical type. The number of muscle fibers in motor units range from 200 to 300 in hand muscles to more than 1,000 fibers per motor unit in the large muscles of the lower extremities. Over the past several decades, the properties of single motor units in skeletal muscle have been extensively examined. Nearly all motor units in the muscles studied thus far can be described into one of three major types. Table 3.3 shows some of the common characteristics of three major classes of motor units. The presence of varying proportions of motor units of different types of different type provides muscle with the ability to respond appropriately to postural and locomotive demands. Muscles that are required to produce moderate to low levels of tension along periods of time contain a high percentage of fatigue-resistant muscle fibers. Muscles required to produce quick, high-force levels of brief intervals contain a higher percentage of strong, fast-twitch, fatigable units. Control of force, generation, and volitional contraction. Studies of voluntary contraction in humans have demonstrated that two main processes are used by the central nervous system to regulate the force output of skeletal muscle. To initiate contraction in particular muscle, the CNS must first excite alpha motor neurons that innervate the muscle fibers. The number of motor neurons and hence the number of motor units activated is one primary determinant of the level of muscle contraction produced. Recruitment is the term used to describe the process of increasing the number of motor units brought to excitation to produce increasing levels of muscle contraction. Precisely which motor units are recruited is determined by both depending CNS and peripheral reflex inputs to the alpha motor neurons. If excitatory inputs to the motor neurons sufficiently overwhelm inhibitory inputs to these cells, action potentials will be produced in the neuron's axon and will evoke contraction. One question that faced neuroscientists for years was, how does the CNS know which motor neurons to activate in order to produce a particular level of contraction? Available evidence now indicates that motor neurons are recruited in most contractions in an orderly sequence. The CNS command to begin muscle contraction first activates the smallest, highest internal resistance alpha motor neurons. If more focus is required to properly perform an activity, The command signals from the CNS are increased, and progressively larger, lower intestinal resistance motor neurons are activated. The recruitment order of motor units into both reflex and voluntary contractions depends on the size of motor neuron cell body. The small motor neurons require less synaptic current to excite them sufficiently to produce action potentials. As the size of motor neuron increases, greater amounts of synaptic current are required to excite these cells. The size-dependent recruitment of motor units into contractions is now commonly referred to as the size principle. Because the size of alpha motor neuron is related to the type of muscle fibers innervated by the neuron, recruitment of motor units in voluntary contractions will generally follow from type S, slow twitched fatigue resistance motor units, into type FR, fast twitch fatigue resistant units, and finally to type FF, fast twitch fatigable units, as the level of contraction increases. Those motor units designed to generate tension for relatively long periods without substantial fatigue, type S and FR, are therefore used most in volitional contractions. Type FF units, which are capable of producing high tension levels for very short periods, are used only occasionally in high force level contractions. The second way in which the nervous system regulates muscle tension generation is by controlling the rate of motor unit discharge during recruitment. This control process is commonly referred to as rate coding. If higher levels of tension are required as contraction is proceeding, the CNS may simply increase the discharge rate of the motor units already active in the contractions. As described previously, increasing the firing rate of motor units increases their force output as summation of contraction. Discharge rates of motor units in human voluntary contractions begin at about 5 to 10 per second and rarely exceed 30 per second. During voluntary muscle contraction, motor units are recruited in an asynchronous manner. That is, motor units are not all activated at the same time. This asynchronous activation has the effect of smoothing the tension output of the whole muscle, even when the individual firing frequencies of motor units may well be below the level at which fused tetanus would occur for the individual units. Discharge frequencies of motor units recruited in voluntary contraction are also not all the same. Some units may be discharging at low steady frequencies, while others may discharge irregularly at higher or lower frequencies. The relative importance of recruitment and rate coding in the regulation of muscle tension has not been clearly established. Some evidence suggests that in muscles of the hands, which often produce delicate controlled movements, rate coding may be a predominant mechanism by which force is regulated. In contrast, in the large muscles of the lower extremity, recruitment may be dominant mechanism regulating the level of contraction. Clinical electrical stimulation of nerve and muscle. In the intact peripheral neuromuscular system, the application of electrical stimulation can induce action potentials in nerve and muscle that are indistinguishable from those evoked by the normal action of the nervous system. Furthermore, action potentials evoked by electrical stimulation in peripheral alpha motor neuron axons elicit contraction of skeletal muscles that appear to be the uninformed person to be identical to voluntary contraction. In fact, muscle contraction in response to electrical stimulation is very different from that produced by normal physiological mechanisms. One of the objectives of the remaining sections of this chapter is to identify the similarities and differences between the nature of contraction in response to electrical stimulation and normal physiologic activation. Electrical stimulation may also activate peripheral sensory nerve fibers and the nerve fibers of the autonomic nervous system. The electrical stimulation of sensory fibers gives rise to the characteristic tingling sensation associated with such stimulation and has been found useful in management of a variety of pain syndromes. But it does not produce patterns of sensory action potentials like those generated under physiological activation. Activation of excitable tissues with electrical stimulation. How does the induction of current in biological tissues bring about the activation of nerve or muscle? Figure 3.18 shows two electrodes of a stimulator circuit applied to the skin directly over a small branch of a peripheral nerve. As the stimulation is initiated, one electrode briefly contains excess of negative charge, and the other electrode is deficient in negative charge. Ions in the region will migrate toward or away from these electrodes according to their charge. The pattern of current induced by the movement of these ions is represented by the lines connecting the electrodes. Some of the ionic movement occurs in the extracellular fluid, and some of the current passes through the nerve membrane. The net effect of this current is slightly depolarization of the nerve membrane. If the brief current induced across the membrane is very small, the change in the transmembrane potential will rapidly return to resting membrane potential. If the current induced across the membrane is large enough, voltage-gated sodium and potassium channels will open, and an action potential will be evoked and propagated along the membrane. The evoked action potential is identical to that produced along the fiber membrane in response to physiological activation. Action potentials evoked in peripheral nerve are transmitted along the nerve fiber in both directions and from the point of initiation. There is conduction in the direction of normal propagation, orthodromic, as well as action potential propagation, and the opposite direction, antidromic. Antidromic conduction is toward the periphery in the sensory nerve fibers and toward the CNS in motor nerve and autonomic fibers. Stimulus characteristics for activation of excitable tissues. The current induced in biologic tissues must be of sufficient amplitude, strength, and duration to bring excitable cells to a critical transmembrane voltage above the resting membrane potential, called the threshold, in order to evoke an action potential. For a particular single excitable cell, there is a family of stimulus strength, duration, or SD combinations that can bring the cell to the level of depolarization required to trigger the opening of voltage-gated sodium and potassium channel proteins. Plotting the SD combinations that are sufficient to activate an excitable cell and connecting the points yields a strength duration curve. Any stimulus SD combination that falls below or to the left of this line will not initiate an AP and is described as subthreshold. Those stimuli with SD characteristics above to the right or the curve are called suprathreshold and are always adequate to activate the fiber. For a particular type of excitable tissue, the minimum amplitude of stimulus that will activate the tissue at very long stimulus durations is called the Rio base. The minimum duration of a stimulus at twice the Rio base, that is just sufficient to activate the excitable tissue, is called the chronoaxis. In the past, electrical stimulators were used clinically to determine the Rio base and chronoaxis values for the peripheral neuromuscular system. The reliability of such techniques is poor. Today, sophisticated electrophysiological evaluation procedures such as nerve conduction velocity studies and electromyography are commonly used to assess peripheral nerve and muscle integrity. Peripheral nerve fibers are inherently more excitable to electrical stimulation than muscle fibers. Consequently, stimuli with the ST characteristics sufficient to activate peripheral nerve fibers will not be strong enough to activate isolated muscle fibers. Stimuli must have larger amplitudes and longer durations to initiate APs and denervated muscle fibers. Figure 3.20 shows the SD curves for the 3 type A peripheral nerve fibers and isolated muscle fibers. Because nerve fibers contained within a peripheral nerve are not identical in diameter or internal resistance, the relative excitability of these fibers to electrical stimulation varies. When a mixed peripheral nerve is directly stimulated, those fibers with the largest diameter and lowest internal resistance are the most easily excited. When electrical currents are produced in peripheral nerve, the AA group of nerve fibers are those most likely to be activated based upon their inherent excitability alone. To activate group A, B, or AD, or group C, fibers require stimuli of progressively larger amplitude and/or duration, a process by which increasing numbers of nerve fibers are activated by progressively increasing the amplitude and/or duration of the stimulus is called fiber recruitment. For isolated motor nerve stimulation, the pattern of recruitment tends to be in an order from largest diameter fiber to smallest diameter fiber. In peripheral nerves, which is a pattern opposite to that occurring during volitional activation. In most clinical applications of electrical stimulation to evoke muscle contraction, surface electrodes are used. In such cases, the tendency to recruit larger diameter axons before smaller diameter axons is weak. In fact, a compelling argument has been made that in a clinical neuromuscular stimulation, the recruitment pattern of peripheral axons and hence motor units is non-selective. That is, clinical neuromuscular electrical stimulation recruits both smaller and larger diameter alpha motor neuron axons and hence recruits both the smaller, slower contracting and larger, faster contracting motor units simultaneously, regardless of the relative magnitude of stimulation. The effects of inducing currents on nerve and muscle fibers depend not only on the inherent excitability of these tissues, but also on their location with respect to the electrodes used to transfer the current. In general, the closer the excitable tissue is to the electrodes, the more likely it is to be activated by the current. At a fixed intensity of stimulation, smaller diameter axons very close to the stimulating electrodes may be activated before large diameter fibers located further away. As a result, AB touch pressure sensory nerve fibers near the skin are commonly recruited before the more inheritable A motor and sensory fibers. Because the distance between the electrodes and target tissue influences the precise nature of the activation, it is important to position the electrodes as close as possible to the target tissue to achieve optimal activation. Because both the inherent excitability of the nerve fibers and the location influence the order of nerve activation. When ES is applied over a mixed peripheral nerve containing motor, touch pressure sensory, and pain sensory fibers, ES will normally evoke a pins and needles sensory response before either motor or painful responses. If the amplitude or duration of stimuli is increased sufficiently, motor responses will be produced and superimposed on the sensory stimulation. If stimuli amplitudes or durations are increased even more, evoked ES may evoke a painful response. Electrical stimulation may evoke a painful response, which occurs simultaneously with the sensory and motor responses. In most clinical electrotherapy applications, the pins and needles sensations produced by the stimulation of cutaneous touch pressure afferents will precede motor or painful responses. If stimulation is applied in a region where no alpha motor neuron axons or skeletal muscle fibers exist, for example, over bony prominence, an increase in stimulation amplitude will likely produce a painful response in the absence of muscle contraction. Another important consideration in applying electrical stimulation to activate muscle and nerve is the polarity of the electrodes. This is especially important in the use of monophasic pulsatile currents or asymmetrical biphasic currents. With monophasic waveforms of fixed amplitude and duration, for example, the negatively charged electrode, cathode, is usually slightly more effective in activating excitable tissue than the positively charged electrode, or anode. In the past, when the initiation and cessation of a single pulse were not electrically controlled, pulse durations were relatively long. The application of fixed amplitude current to motor nerves in this fashion revealed that contractions could occur in response to both closing the stimulating circuit and opening it. In addition, contractions could be elicited by either cathodal or anodal stimulation. Overall, the response to the cathode upon closing the circuit, cathode closing current, or CCC, was most effective approach to neuromuscular stimulation. The contraction response evoked by the anode upon closing the circuit was the next strongest, followed by the contraction beneath the anode upon opening the circuit and the cathode upon opening the circuit, respectively. The relationship between the anodal and cathodal opening and closing currents has been called ERB's polar formula. With the very short duration pulses used today in electronically controlled stimulation, contractions are not elicited by opening currents during neuromuscular stimulation. Clinical responses to nerve and muscle stimulation. Sensory level stimulation. When electrodes are placed on the surface of the skin and the amplitude of electrical stimulation is gradually increased, three general forms of response are consistently noted in normal individuals. At relatively low amplitudes, the first response reported by patients is sensory. Commonly, patients identify a pins and needles tingling sensation if the frequency is greater than a few pulses per second, or a tapping sensation if the frequency is set to 1 to 2 if the frequency is set at 1 to 5 pulses per second. This sensory level stimulation is not uncomfortable to most patients and results from excitation of those sensory nerve fibers that lie in or near the skin in the vicinity of the electrodes, where the current density is greatest. As the stimulation amplitude is gradually increased, the sensation becomes stronger and often spreads to the region between the electrodes and deeper into the tissues. As can be observed from Figure 3.22, sensory level stimulation can be achieved by the use of many different amplitudes and duration combinations. Whether sensory fibers are activated is independent of stimulation frequency selected. If sensory level stimulation at frequencies greater than about 15 pulses per second is maintained for prolonged periods of time, the subject will generally note a gradual diminution in the ability to sense the stimulation, a phenomenon known as adaptation. Adaptation may be reduced by intermittently interrupting the stimulation or by varying either in amplitude or frequency. Motor level stimulation. As the intensity of stimulation is gradually increased, the tingling sensation felt by patients increases as progressively greater numbers of sensory nerve fibers are recruited. In addition, the activation threshold of alpha motor neuron axons lying in peripheral nerves innervating skeletal muscle is soon reached. The excitation of alpha motor neurons axons is in the intact nervous system that produces muscular contraction is called motor level stimulation. At low levels of motor level stimulation, contraction may only be detected by palpation of innervated muscles as the amplitude or pulse duration is increased. The contraction soon becomes strong enough to produce visible joint movement. The form and strength of muscular contraction produced by ES are determined by the same two processes, recruitment and rate coding, involved in voluntary contraction. There is progressive activation of great numbers of alpha motor neuron axons as amplitude of stimulation is increased. Motor neuron recruitment increases the number of muscle fibers activated and hence increases the force output of the stimulated muscle. In voluntary contraction, motor units are recruited from smallest to largest as the requirement for force are increased. In stimulated contraction, recruitment was thought to occur from largest to smallest as the stimulation strength is gradually increased. Stimulated contractions were thought to occur by an activation of type FF motor units first, followed by type FR units and ending with type S units. As indicated previously, this reversed order of motor unit recruitment in electrically induced contractions does not appear to be supported by recent research. Rather, it appears as though surface electrical stimulation recruits a combination of FF, FR, and S motor units simultaneously. Recruitment in stimulated contractions is controlled by varying either the magnitude or duration of each stimulus or some combination of two stimulation parameters. Increasing either amplitude or pulse duration increases the phase or pulse charge of each stimulus, increasing the activation punch in each waveform. Figure 3.2c shows the relationship between the amplitude of fixed duration stimuli delivered at a tetanic frequency and the resultant contraction force of activated muscle. The graph shows that after the motor threshold is exceeded, very small increases in stimulation amplitude produce relatively large increases in the force of muscular contraction and recruitment increases rapidly. During the use of electrical stimulation to activate muscle, clinicians must exercise caution in the adjustment of stimulation amplitude to avoid undesirable marked increase in the level of muscle contraction. On initial exposure to electrical stimulation for muscle activation, clients frequently exhibit a low tolerance to the stimulation. As a consequence, evoked muscle contraction may not be sufficient to produce either physiologic changes or appropriate joint movements. Repeated exposure to electrical stimulation is generally accompanied by an increase in client tolerance and associated increases in the force of evoked contraction. Increases in the stimulation amplitude may be possible not only from the session to session, but also between individual contractions within the same treatment session. The second primary mechanism regulating the force of contraction involuntary contraction, rate coding, is also primary mechanism in regulating stimulating contractions. If the stimulation frequency is low in the range of 1 to 5 pulse per second, elicited contractions will be twitch-like in character. If the frequency of the stimulation is gradually increased, partially fused contractions are formed, followed by fused tetanic contractions as the stimulation frequency rises above the range of 25 to 30 pulses per second. As involuntary contraction, as the frequency of motor unit activation rises, the force output of muscle rises. Another key difference between voluntary and electrically induced contractions is the synchronous activation of all motor units in the stimulated contractions. The initiation of thick and thin filament interactions occurs in all recruited fibers at about the same time. The previous discussion reveals that electrically elicited contractions are not truly physiological contractions. In spite of this fact, electrical stimulation can be used clinically to produce physiological changes within muscle for strengthening and functional movement, which may allow individuals to function better in their daily activities. Both isometric and isotonic contractions can be produced by the electrical stimulation of muscle. Isometric contractions can be produced by physically blocking the movement about a joint, or by simultaneously stimulating both the agonist and antagonist acting on a joint. Isotonic movements can be evoked by electrical stimulation when the joint on which the stimulated muscle acts is not stabilized. A common application of an isotonic movement produced by stimulation is ankle dorsiflexion during ambulation to assist in the swing phase of gait. Such stimulation is frequently triggered by a special switching device placed beneath the sole of the foot. In eliciting isotonic contractions, the user might realize that very strong isotonic contractions to the fully shortened position of a muscle may produce severe cramping in the core of the stimulated muscle, and hence such contractions should be avoided in most therapeutic applications. The application of an uninterrupted train of pulses at an amplitude high enough to elicit muscle contraction will very rapidly induce muscle fatigue. To combat this potential problem in clinical applications, stimulator trains are commonly interrupted at prescribed intervals. When the stimulation is adjusted to a 1:1 on time to off time ratio, e.g., 10 seconds on, 10 seconds off, muscles tend to fatigue rapidly. In contrast, 1:5 ratios, 10 seconds on, 50 seconds off, tend to significantly reduce the onset of muscle fatigue. Noxious level stimulation. The final level of clinical electrical stimulation is that which is painful to the patient and is referred to as noxious level stimulation. In this situation, the intensity of stimulation is so high that many A, D, and C nerve fibers, which normally carry signals associated with painful stimuli, are activated. Action potentials produced in first order nociceptive afferents in turn activate the spinothalamic pathways leading to the somatosensory cortex and result in the perception of pain. Because the level of stimulation required to activate the small diameter pain efferents is generally very high, strong muscular contractions often accompany this form of stimulation, and motor neurons are located close enough to the stimulus electrodes. If noxious stimulation is carried out in a region where no motor axons or muscle fibers are present, e.g., over a bony prominence, a painful response may be elicited without the production of any muscular contraction. The precise stimulation parameters that evoke a painful response will vary from individual to individual. Other factors, such as electrode placement, stimulation waveform, and type or location of sensory fibers in the region of stimulation, will also influence the level of stimulation that evokes a noxious response. This chapter has reviewed the central features of nerve and muscle morphology and physiology to provide a foundation for understanding the clinical application of therapeutical electrical stimulation. In addition, volitional and stimulation-induced muscle contraction properties have been compared and contrasted. Specific techniques commonly used in clinical settings for the management of the selected disorders are presented in subsequent chapters. More detailed information on the structure and function of nerves and muscles can be found in comprehensive references and reviews. Astute users of electrotherapy must remain sensitive to the fact that the results of ongoing research and clinical studies must be integrated into their knowledge. Such new information may prompt significant changes in the application of electrical stimulation to obtain optimal therapeutical benefit. And that ends Chapter 3, Physiology of Muscle and Nerve.